So next, we're going to move to a forward genetic approach to sleep genes in humans. Dr. Ying Hui Fu is a professor of neurology at the University of California, San Francisco.
Okay, so our research team, our goal is really try to get a better understanding of what is optimum sleep for human and how is it regulated, so we can come back to help people, hopefully, sooner rather than later. And so today what I'm going to do is I'm going to use 3 examples to show you some of the advantages that we can have by using a genes first approach. In our case, we use it on human sleep behavior trait. So over the last 25 years, our research group mainly focused on 4 different human sleep behavior traits, including morning larks, night owls, and natural short sleepers. These are people who naturally just sleep fewer hours, and natural long sleepers. And today I'm just going to talk about 1 example for the morning lark and 2 examples for the short sleepers. So of course, these 4 different sleep behavior traits can be divided into 2 groups. One group is about our sleep schedule, and that's all about what is the best time for us to go to bed, what is the best time to get up, that would be best for our total well-being. And the other group is about sleep duration, and that's about how many hours of sleep we should get that would be best for our health and also for our cognitive function. So for the sleep schedule, over the last 20 years, we have reported more than a dozen mutations. And all these genes that reported so far are on these molecular core clock components that Dr. Herzog talked about 20 minutes ago. And the interesting thing I want to point out here is that some of the mutation carriers for some of these genes actually, in addition to have in addition to be morning larks, they also have other conditions. For example, the mutation carriers for PER3 gene, in addition to be a morning larks, they also have seasonal affect disorder. So by studying some of these genes actually can help us understand the connection between sleep and some of these additional conditions. So what I'm going to do today is just to use the example, the mutation that we found in the gene called PER2 as an example. So PER2, as Eric said, is part of the molecular core clock components. So the production of the PER2 protein is under circadian regulation. And I'm just going to tell you what we have learned, give you a summary because of the time. So the mutation was found in a serine amino acid change to glycine. So when we were studying these mutations, we learned that the serine is modified by phosphorylation, which is one kind of post-translational modification. And we know that post-translational modification on proteins are very important because it kind of tells proteins what to do next, right? So when we were studying this, we learned that the phosphorylation on this side and many other phosphorylation sites and many phosphorylation sites on many proteins are under circadian regulation. So not only protein production is under circadian regulation, the post-translational modification is also under circadian regulation. So that you can, this is another example of why circadian body clock is so important for our total well-being. So we found that there are 5 serines in this mutation region. It forms a cluster. They can all be phosphorylated. And interestingly, the serine residues can also be modified by another post-translational modification called O-gluconosylation. So from our study, we learned that 3 out of these 5 serines can be modified by O-gluconosylation. And O-gluconosylation level actually is dependent on our nutrient intake, what we eat, what we drink. So we then learned from our experiment that under high glucose concentration in the cell or body, actually the O-gluconosylation becomes dominant and block off all other phosphorylation and then lead to a shorter period. And this is then lead to a faster clock. So now I want to next talk about sleep duration. So here, how do we study sleep duration? I'm talking about normal people's normal sleep duration, not have nothing to do with the environmental factors. So how do we study this then? Actually, we're taking advantage of this group of people. We call them natural short sleepers. And these are people either lifelong or most of their life only need 4 to 6 hours sleep a day and stay very active during waking hours and live a long, healthy life. So since 2009, we published 5 mutations on 4 genes for this natural short sleep behavior trait. And what I'm going to do today is use the ADRB1 mutation as example to show you what we have learned with some of these genes about the circuitry for regulating sleep duration. So for ADRB1, we found that ADRB1 neurons in the brain region, dorsal pons region, or peri-tegmental reticular nucleus, or peri-TRN region, both of these 2 regions are part of the brainstem. So in both of these 2 regions, ADRB1 neurons are active during wakefulness and REM sleep, and very quiet during non-REM sleep. However, if we activate these neurons in the dorsal pons, we can only increase wakefulness or wake them up from non-REM sleep, but not from REM sleep or wakefulness. But in the peri-TRN region, if we activate this neuron, we can increase their wakefulness from wake, REM, and non-REM, all 3 states. Now, we also studied ADRB1 neuron in the central amygdala region and found that in the central amygdala region, ADRB1 neurons only affect REM sleep. It does not affect wake or REM sleep. Now, if we just focus on the peri-TRN region, and there are actually subgroups of neurons, and one group project to lateral hypothalamus. Now, if we stimulate the end of the projection in the lateral hypothalamus, we found that they behave very similar to the cell body in the peri-TRN. They can wake this animal up from wake, REM, and non-REM. And there is another group of neurons that project to parafacial zone, actually, in addition to increased wakefulness, also increase REM sleep. So I think in summary, what we learned from ADRB1 neurons circuitry study, and we're also learning this with a different gene and different mutation, is that the circuitry for sleep duration or sleep quality regulation is quite complex. And so the third example I want to talk here is about sleep and healthy aging. And I think we have heard many examples today that poor sleep can contribute to health problems. And so here, we were wondering whether we can look at the other side of the coin. And so this is an example for that. So we were studying these natural short sleepers for a number of years. And after a few years, it was very curious for us because on one hand, we knew that we, human, if we subject ourselves to long-term sleep deprivation or disruption, we have much higher chance to get all kinds of diseases from high blood pressure all the way to Alzheimer's or cancer or whatever. On the other hand, we have this group of short sleepers, natural short sleepers, they lifelong sleep 4 to 6 hours and remain very healthy and live a very long, healthy life. So after a number of years, it occurred to us that most logical explanation for this will be that actually these short sleepers, even though they sleep fewer hours, their sleep quality or sleep efficiency in terms of performing whatever function sleep is doing is more efficient, is higher. And because this better sleep quality and better sleep efficiency, then offer them a protection from all kinds of disease of aging. So we decided to test this idea. So what we did was that we put the short sleep mutation onto 2 different kinds of Alzheimer's mouse model. One kind forms A beta plaque, it's 5XFAD. The other kind is from the tau tango type called PS19. So far, what we have done is that we have put 2 different natural short sleep mutations onto 5XFAD mice. The DEC2 here, if you see DEC2 wild type control crossed with 5XFAD, you see a lot of plaques here. And the mutant DEC2 crossed with 5XFAD, you see dramatically decreased plaques. Now 5XFAD control, again, a lot of plaques, but when we put MPSR1, the second mutation, onto 5XFAD, again, you see dramatically decreased plaque. Now with the tau tango mouse model, we cross JAK2 mutation, ADRB1 mutation, and MPSR1 mutation, 3 different short sleep mutations onto this mouse model. Here you see PS19 control mice with a lot of tau tangle, with the green indicated tau tangle, and with the JAK2 mutant, ADRB1 mutant, and MPSR1 mutant, you see that dramatically decreased tau tangle. So with these 5 examples, I think we have shown a strong evidence that these short sleep mutations seem to offer this human protection against Alzheimer-like diseases. And we believe this is going to be true for many other diseases as well, because this human, we see from this human, they actually live a long, healthy life and don't seem to have any health problems. So with that, I think I will stop there.
Wow. Thanks, Ying Hui. That was fantastic. And you matched the fabulous timing of Dr. Raisin.